So good morning. My name is Suresh Kuchipudi. I'm a clinical professor and associate director of the Penn State Animal Diagnostic Lab. And today I'm going to briefly give an overview of the research that we are doing currently that focuses on human and animal health from COVID. And as we all know, the coronavirus disease 19 pandemic is the greatest public health catastrophe in the modern era. We never saw a infectious disease pandemic of this scale in over 100 years now. And what this pandemic has done is that it spread a completely new virus called as severe acute respiratory syndrome coronavirus 2 across the world at an unprecedented scale. And as this is a brand new virus, we did not have any information about this virus as we entered into this pandemic. And in the past 8 to 9 months, the scientific community globally has been doing a lot of research to understand the nature of this virus and answer a lot of questions. And we still have many questions to be answered. So one of the fundamental questions is, where do the coronaviruses actually live? Do they come out of nowhere, or what is the source of these viruses? And based on the scientific research, we know that the coronavirus, the term implies a group of viruses, not just one member, hundreds of them. They adapted to virtually live in most animals and species and humans over a period of perhaps 50,000 years or even longer than that. And the way these viruses adapt is that they typically tend to remain in the species that they are settled in. And occasionally, they do also learn this art of jumping out of the species, what we call a spillover event, and infect a completely new host and start spreading in that new host. And that is how, if an animal virus learns this skill of infecting humans and jumps out of humans, starts spreading among humans, that will result in an epidemic. And if it becomes widespread, it will result in a pandemic. And the question is then, why are these viruses doing this, or how are these viruses are able to do this feat of jumping out of species and infecting a completely new host? It is because of the nature of the virus itself. So my lab particularly studies RNA viruses, so the viruses that have RNA as their genetic material. So as you may know, that most higher forms of life have their genetic information written on deoxyribonucleic acid called as DNA. And viruses that have RNA as their genomes, when they replicate, meaning when they are making babies, they have to copy their genome multiple number of times. And in the process, they make mistakes. And they lack a mechanism that can correct these mistakes. So as these mistakes, scientifically called as mutations, accumulate over a period of time, then the virus can gain an ability to do something completely different than the parent virus was ever able to do. And that's what contributes to the cross-species transmission of these viruses. So in terms of human epidemics, typically a coronavirus that lives in the wild animal reservoir like bats or rodents, where researchers have found hundreds and hundreds of completely new coronaviruses that we don't know the relevance of. At some point, they gain the ability to infect a totally different animal. It could be a domestic animal or a wild animal. The first step, which is called as interspecies transmission. And after some time, when the virus is circulating in these animals, because of the close proximity of animals and humans in certain parts of the world that will facilitate the constant exposure of humans to such animals, the virus at some point will then make the next step of jump to start infecting humans. And after some time, the virus will learn the skill of effectively infecting humans and then transmit among humans, and that would result in an epidemic or a pandemic. And this is precisely what we believe has happened with the two previous coronavirus epidemics in the 21st century, the SARS in 2002 and the Middle Eastern Respiratory Syndrome coronavirus disease in 2012. However, the precise origins of the COVID-19 virus are still being debated, and we need to have more research to have solid scientific backup for any theory. But based on the science at this point, what we believe is that the virus originally came out of bats. However, the intermediate host, or is there an intermediate host, is still being investigated, and the possibility of an animal model called pangolins is proposed. But however, we do know how the virus eventually spread globally. So by community contact and hospital-borne infections, and more importantly, due to the global movement of people, the virus was carried to different countries through infected people. And this is particularly relevant to highlight the fact that in COVID-19, unlike the previous viruses, the incubation period, which is the time when somebody is exposed to the time when they actually start showing symptoms, there is a gap. And during this period, the people who may have exposed but have no idea that they're exposed could be unintentionally spreading the virus to other people. And that is the most critical aspect in the spread of the virus. And based on CDC's numbers, we believe that at least 50% of the transmissions happen through people that are either not showing symptoms or yet to show symptoms. So that is a critical piece to keep in mind. And the pandemic has now very close to the causing 7 million cases in the US, recorded number of cumulative cases. And the deaths cumulatively have crossed this grim milestone of 200K deaths, and the virus is still spreading. So ever since the pandemic started, my lab has switched gears, and to respond to this critical need, we have been doing a lot of different research aspects. And I'm going to highlight three major projects in this area that are relevant to animal and human health. So in order to do this research, one of the incredible opportunities we have at Penn State is the expertise, but more importantly, infrastructure that would allow us to quickly respond to these. And one of them I wanted to highlight is the Biosafety Level 3 lab, which will allow us to study this dangerous pathogen safely in a very, very high containment setting. As you can see in the picture here, the researchers would wear what appears like a space suit so that they can be safe and handle these viruses. So this allowed us to respond and create these important research projects in a very short span of time. So the first project is the question that what is the impact of COVID-19 to animal agriculture? And the association of animals and humans in coronavirus is widely known. And especially the previous SARS coronavirus that has only spread to 28 countries and infected less than 9,000 people, we have evidence that the virus has indeed spilled into pigs. So now this virus has spread at an unprecedented level that we never saw a coronavirus do before. And we already have evidence for transmission of the virus from humans to certain animals, including tigers, lions, dogs, cats, and ferrets. Although we don't have evidence for the virus transmitting from these animals to humans. And an important question is then, what is the, given the widespread nature of the virus among humans, agricultural animals are also being exposed to the virus, conceivably. So the question is then, can the virus infect these animals? And the second relevant question is, if it can infect these animals, then can the virus then become efficient in spreading among these animals? So in order to be proactive and understand before this can happen, so we could develop better mitigation strategies. We put together a team of multidisciplinary scientists. My lab, along with chemical engineering and College of Sciences at Penn State, we received the competitive grant from USDA to do this combination of computational and infection studies to really ask this question: can agricultural animals be infected by this virus? And what are the chances of this virus becoming more efficient to spread among these animals? And this is really critical because before we wait for this to happen, it's always efficient to be proactive and ask this question so we can be better prepared and minimize if there is any possibility of losses to the animal agriculture by this virus. The second project is the development of a novel COVID-19 vaccine. This is a study funded by the National Institute of Health. And we all know that in the end, an effective and safe vaccine is what is needed to end this pandemic. And I would like to highlight that the previous research findings from the earlier SARS coronavirus has done a great help to give us head start in developing COVID-19 vaccine. There are several vaccines that are developed and currently in clinical trials. However, there is a need to be a bit more proactive if these pandemics or epidemics were to happen in the future. Could we develop platforms that can rapidly be deployed in the event of an outbreak that have a known safety profiles? So with this in mind, our approach, which is a collaboration with Purdue University and CDC, is to use a virus from cattle, and we, by genetic modification, we render the virus harmless by removing the disease-causing parts of the virus, and essentially use the virus as a nano vehicle, and then we then engineer the spike protein which is needed for the immune response in humans from the SARS virus, and that gene gets inserted into this virus along with other immunogenic peptides, and which can then be safely used to vaccinate humans that can then develop resistance to the infection. And the advantage with this platform is once we prove the efficacy and then the safety profiles, and it can be a very effective plug-and-play type device. If we have a new pandemic virus or epidemic virus, this can then be quickly deployed to make an effective vaccine against that particular virus. And finally, the project I'm going to talk about is the development of smarter COVID diagnostic tools. And again, we all know how critical is testing to better understand the pandemic, but more importantly, to minimize and stop the spread of the virus to other human beings. And currently, the lab testing methods are complicated, and they require very sophisticated infrastructure and highly trained personnel. And it also takes at best 1 to 2 days, and sometimes if there is demand, the turnaround time can be as high as 5 to 7 days. And these methods inherently, as they require highly sophisticated equipment, they are not very easily scalable. So to address this need, we teamed up with engineering and biomedical engineering scientists in Penn State, and this study is funded by the National Science Foundation. So the idea is to develop a test that can be handheld and can be used as a point of care test, but this is accurate and can detect the virus in less than 5 minutes. And this uses a complex biophysical features to basically tag the virus to magnetic nanoparticles that are tagged to fluorochrome. So the virus can be visualized in real time in less than 5 minutes. Finally, I would like to thank the members of my lab who does this incredible amount of work. And thanks also to all the funding agencies and our college administration for the excellent support. And I thank you very much for your attention, and I'll be happy to answer any questions.